Hi everyone, welcome to my channel Capsa. So today's topic is about staining technique. What are the different type of staining technique? So why staining is necessary? So it is necessary because first of all to produce color contrast, second to increase the visibility of the object or the specimen which we are examining under the microscope. Now before staining, the fixation of smear must be done. It can be done in two ways. First is heat fixation. It means by using gentle flame or heating, and after that air drying. Okay. And chemical fixation, second method, it means by using chemicals such as ethanol, acetic acid, formaldehyde, etc. And chemical fixation actually useful for the examination of bloods. Okay. Now what are the staining methods? These are mainly four types. First one is simple staining, second one is negative staining, third one is differential staining, and fourth one is other staining. Okay. Now we will understand everything clearly. So the first staining method is simple staining method. It means in this case directly staining of the bacterial cell is done with the using of positively charged dye. Okay. So basic colors such as methylene blue or fuchsin is used, and it used for the examination of or identification of morphology and the structure of bacteria. Okay. Now what are the steps? So the steps are, first one is air dry the slide containing bacterial smear. It means you need to prepare the bacterial smear by using inoculating loop or inoculation loop and then air dry the slide. Then in the second step, flood the slide with stain. After few seconds, wait for few seconds to fix the stain with the smear. Then after few seconds, wash the slide. Then air dry the slide. And after final step, after air drying, observe the slide under microscope. That is simple staining. Now, what is negative staining? So, negative staining actually background gets stained in black color or any kind of color. It can be ash or dark. Means under dark background, unstained bacteria are observed, which is more visible due to the contrast. So which kind of color are used? First one is Congo red or second one is Nigrosin. These two colors are used in negative staining. And this type of staining method actually used for the examination or identification of bacterial capsule, which do not take up the simple stain. That's why negative staining is used. Okay. Now what are the steps? So in the first step, stain is taken into the slide. Obviously, you need to prepare the smear, then stain is taken into the slide. Pick up small amount of sample and stir it with the color. It means the stain and the smear or the sample should be in a mix. Okay, then air dry the slide. Do not heat fix. After that, flood the slide with acid alcohol, which composed of 95% ethanol and 3% acid. In the fourth step, flood the slide, in the fifth step, drain the excess acid alcohol. Do not wash, obviously. Then allow the slide to air dry and examine under the microscope. In this step, the stain and the sample should be mixed up. Then air dry the slide. Clear? Then use decolorizer, means acid alcohol. Then air dry the slide and examine under the microscope. That is negative. So after negative staining, bacillus species are visible like this. So in simple staining and negative staining, what is the basic difference? In simple staining, we use the basic dyes. which obviously contain positive charge, and in negative staining we use acidic dyes which possess negative charge. Okay, and we know that bacterial cell is negatively charged. As a result, in this case, simple staining, obviously dye will attract it and the cell will stain. So the cell will stain. But in case of this, the similar charge which repel each other, so the dye repelled the cell, so obviously the cell remain unstained. That is the basic difference between simple stain and negative. Coming to the next, what is differential staining? Okay, so what do you understand by differential staining? In differential staining, two type of stains are used. First one is primary stain and second one is counter stain. Okay, so three categories which comes under differential staining are gram staining, acid fast staining and Albert staining. I have discussed already gram staining. You will get that lecture in my microbiology playlist. Okay, so I will discuss here the acid fast staining. So in the acid fast staining, first of all you need to understand what do you understand by acid fastness. So acid fastness is the ability of a bacteria to resist decolorization with the acid alcohol. That is acid fastness of the bacteria. Okay, and it is actually due to the presence of high concentration of mycolic acid. Okay, high presence of mycolic acid, which is a lipid present in their cell wall. Okay, so primary stain here uses carbol fuchsin. Okay, this is a lipid soluble dye, and the counter stain which is used is methylene blue. This type of stain or Ziehl-Neelsen stain is a kind of acid fast staining, and this type of staining used for the identification of Nocardia, Mycobacterium species, etc. Okay, now we will see what are the steps involved in acid fast. So first air dry and heat fix the smear, flood the slide with carbol fuchsin, that is primary stain. Then steam the slide with Bunsen burner over the sink, because due to steaming of the primary stain will be more soluble and more lipid soluble to the cell wall. Okay, it means steaming will increase the penetration power of the primary stain. Then in the fourth step, let the slide set for 5 minutes and rinse with water. In the fifth step, again flood the slide using counter stain. What was the counter stain? That was methylene blue. And wait for 30 seconds and rinse with water. Then dry the slide by using babulous paper or also known as blotting paper. Then observe the slide under oil immersion microscope. So that are the steps. And after examining, acid fast staining will look like this, and this is babulous paper or also known as blotting paper. Okay. Now next stain is, which is a kind of differential staining, that is Albert staining. So the Albert name comes from Friedrich Albert, who actually developed this technique to identify Corynebacterium diphtheria. Now in this technique, two type of basic dyes are used: Toluidine blue and Malachite green. And this technique used for the identification of metachromatic granule or volutin granule, which is a storage granule, bacterial storage granule. Okay, so these are the bacterial storage granule. Now we need to understand about two type of solution: toluidine blue, Albert A solution and Albert B solution. The Albert A solution consists of toluidine blue 0.15 gram, malachite green 0.20 gram, glacial acetic acid 1 ml, alcohol 95%, that is 2 ml. And Albert B solution consists of iodine 2 gram and potassium iodide solution. You can see. Now how we will prepare Albert A solution and B solution? To prepare Albert A solution, first dissolve these two dyes, toluidine blue and malachite green, these two dyes in alcohol and add to the distilled water and acetic acid. Okay. After adding, allow the stain to stand for 1 day. Then filter and then add distilled water to make the final volume up to 100 ml. Okay. So this solution must be prepared 100 ml by using distilled. And how Albert stain B solution is prepared? Dissolve the potassium iodide in water and then iodine. Add iodine. Okay. Thus, Albert B solution is prepared. Now, what are the steps? To prepare the smear and flood the smear by using Albert A solution first. Then wait for 5 to 7 minutes to settle down this Albert A solution with the smear. Okay. Then drain off the solution and wash the slide by using Albert B solution. Do not use any kind of water. We need to use Albert B solution to drain off and wash the slide. Okay. Then wait for 2 minutes to fix the Albert B solution. Then wash the slide with water and dry the slide by using blotting paper or blot paper. Then observe the slide under oil immersion microscope. Okay. So after identifying, the image will look like this. You can see this dark color globular structures are metachromatic granule or metachromatic granule. Okay. Now, what are the other type of staining methods? Okay. So, other type of staining methods are pore staining, lipid staining, carbohydrate staining, etc. Pore staining can be done using acid fast stain plus malachite green. Lipid staining can be done by using Sudan black stain. And carbohydrate staining can be done by using iodine stain. Okay. So these are the main different type of staining technique you need to study or to understand for the microbiology. Okay. So this is the end of my lecture. If you like my video, please do like, comment, share. Thank you.